Can I start now?
Yes, you can start.
Hello everyone. I'm presenting a paper titled Molecular Mechanism
None.
of
Your presentation should be at the center and full for making the character be enlarged.
It is not enlarged.
Not enlarged. I don't know.
It is on
Your
my screen.
presentation
It
at
is
the center,
enlarged.
but small size. Can you make it large size? I don't know how to. Maybe right.
I can see the full screen.
Okay. Sorry.
The title is Molecular Mechanism of Biogenesis of Apoptotic Exosome-like vesicles
Louder.
and their role as damage-associated molecular patterns. In this paper, it has been found that when the cells are subjected to the apoptotic death, they release some exosome-like vesicles, which induce the inflammation in the cell. Previously, it was believed that uh, the uh, the particles released from apoptotic cell death cannot induce the inflammation, but in here it has been found that the apoptotic exosome-like vesicles released after the apoptotic cell death can induce the inflammation in mouse macrophages by NF-kappa B and P38 pathway. Introduction: the extracellular vesicles are membrane-bound particles released from almost all kinds of cells, and they contain DNA, microRNA, and messenger RNA. And they are classified as exosomes, macroparticles, and apoptotic bodies based on their size. Exosomes originate from endosomes, and their size range from 50 to 150 nanometer, and their specific density range from 1.13 to 1.19 gram per ml. In this paper, it has been found that the sphingosine 1-phosphate, which is a lipid, which is a membrane lipid, is involved in the release of exosomes after the apoptotic cell death. So sphingosine 1-phosphate is produced by phosphorylation of sphingosine, which is caused by sphingosine kinase 1 and sphingosine kinase 2, and it is degraded by S1P lyase enzyme. The sphingosine 1-phosphate is involved in various physiologic and pathologic conditions like proliferation, cell survival, angiogenesis, or immune cell migration, etc. In here, it has been found that the exosomes, apoptotic exosome vesicles, are actually the damage-associated molecular patterns which are released from the dying cell. So, damage-associated molecular patterns.
None.
Yes.
Too fast. Too fast. It is slowly. Okay? Slowly. Okay.
Damage-associated molecular patterns are released from dying cells or tissue, which stimulate inflammation in the surrounding. As I earlier said, it was believed that the DAMs are only released from necrotic cells, but when there is a massive secondary apoptosis, apoptotic cells can also release damage-associated molecular patterns and cause inflammatory or immunogenic response. In this study, apoptotic exosome-like vesicles are identified as inflammatory elements released at apoptotic cell death. When we go to the results, in here, the cells are treated with apoptotic inducing drug called staurosporin, and then the conditioned media is centrifuged at multiple speeds, here like 2000 G, 10,000 G, and 100,000 G. And it was found that the pellet found after when the conditioned medium was centrifuged at 10,000 G and 100,000 G was able to induce interleukin 1 beta activation in the in macrophages. Yeah, and when the cells were treated with staurosporin, the release of the exosome particles was very high when compared to the control cells. And when the EVs collected were checked for the mitochondrial and ER and Golgi body markers, it was found that they were highly reduced or almost not present in them, but the exosomal markers like CD63 and the lysosomal marker LAMP1 were highly present in them. And this is the electron microscopy structure of the apoptotic exosome-like vesicles, and when the release of the exosomes was tracked in the time, it was shown that the CD63 expression was highest at the 36 hour, but the highest activation of interleukin beta was at 16 hours. It shows that the released exosome vesicles have very short half-life. And when it was checked for the specific density, the major fractions were found between 1.15 to 1.17 gram per ml, and it was also confirmed that these fractions were successful in inducing the interleukin 1 beta activation in the treated macrophages. And in this experiment, this figure shows that after a 4-hour treatment of the staurosporin, the multivesicular endosomes start to appear, and the maximum amount is found at 8 hours. And we can also see at 12 hours, the vesicles are being fused with the cell membrane surrounded by vacuoles, and then they are released as apoptotic exosome-like vesicles.
Why the cellular endosome fraction was visualized by TEM? Why? What relationship between the exosome release and the cellular endosome?
Actually, the exosomes are endosome-based particles. They are released from endosomes inside the cell.
I'm sorry?
The exosomes are released from endosome.
What kind of endosome?
Multivesicular endosome, containing intraluminal vesicles.
So you need to explain about that.
Okay.
Our parts all understand about the intracellular process for producing the exosome, but Professor Kim's part don't know about that. Okay, right? You understand?
Yeah.
Okay, next.
The exosomes express CD63, majorly CD63 at the membrane. So for the confocal microscopy, the cells expressing EGFP CD63 were treated with staurosporin, and it was found that after 1 hour of the treatment, spindle-like filaments were formed from the membrane, which were co-localized with the CD63, and after some time, these CD63 particles moved to the perinuclear region, and then at 8 hours they were they fused with the membrane, and they were released from the cell, and after 16 hours they were completely invisible, showing that all the CD63 containing particles have been released from the cell. And then it was tried to find out what kind of pathway these damage associated particles are following to to be released from the cell. For that, conventional exosomes are released by escort complex, and the two main proteins of the escort complex are helix and Rep27A. When they were knocked down from the cell,
Actually, the Alex and reps, what is the function of Alex and rep 27 in the intracellular biogenesis of exosome?
Rep27A uh, uh, helps in the fusion of the endosomes with the cell membrane, where they are further released from the cell.
And Alex?
Helix recruits the escort 3, which is important for the formation of the for the scission of the interluminal vesicle from the cell membrane.
Okay. So, what is the result?
The escort complex escort 3 finishes the membrane invagination, which then matures to the endosome and multivesicular endosome, and then multivesicular endosome fuses with the cell membrane by the help of Rep27A. When they were knocked down from the cell, it was found that they had almost no effect on the exosome release. So it means that there is some other thing, some other part of the cell or membrane that is controlling the release of these particles. And then to find if the sphingolipid is involved in the release of the AEVs, the sphingomyelinase inhibitor and some other lipid inhibitors were tried, and it was found that the AEV release was highly reduced with the sphingosine 1-phosphate inhibitor, showing that the sphingosine 1-phosphate has a role in release of the exosomes. And also the released exosomes showed
I
decreased.
cannot understand. What is the effect of each drugs? In figure 3A, the dying cells are treated with 3 different kind of the inhibitors, right?
Yes.
All the inhibitors working on the lipid biosynthesis, right?
Yes.
So what pathway? For example, GW480, 4869, seranib2, DMS. Do you know?
Exosomes can be released by either by escort complex or escort independent pathway. In escort independent pathway, multiple lipids are involved in the release of
Really?
exosomes.
Escort independent pathway related to the multiple lipid pathway? Really? Really?
Sphingosine 1-phosphate is also one of the escort independent pathway, and also the seranib2 is inhibitor of the ceramidase, and DMS is inhibitor of sphingosine kinase. And when the
One other question: why we try to inhibit the lipid biosynthesis during the cell dying in the cell dying cells? Why?
Because we already found that the escort complex is not involved in the release of exosomes from the apoptotic cells. So we hypothesize that some lipid or some
independent pathway.
escort independent pathway can be involved.
Until 2018, what is exactly the known as escort independent pathway?
One of the pathways,
One of
ceramidase.
pathway? One? Maybe not. around the 2015 or the 17, I cannot remember. Until 2018, only one the escort independent pathway is known published at the Science Journal. Only one is that the ceramide dependent pathway. Ceramide. Until now, recently I summarized the exosome biogenesis pathway. For that reason, I reviewed the older paper about the biogenetic mechanism of exosome biogenesis. Until we can only divide the one mechanism, ceramide mechanism, as escort independent mechanism. You understand? So until now, the only one pathway about the escort independent pathway is only ceramide. What is ceramide? How the cells produce ceramide in the cell? How?
That I don't know.
You don't
About
know?
ceramide.
Actually, the three drugs in science paper, they found the GW 480, 4869, the science research in science paper, they found GW 4869 block the ceramide dependent exosome biogenesis, completely blocked. So the GW 4869 is inhibitor of sphingomyelinase. And then seranib2 is an inhibitor of ceramidase. And DMS is an inhibitor of sphingosine kinase. Actually, from the membrane sphingolipid, sphingomyelinase making ceramide. And then seranib2, the transform ceramide to sphingosine. And sphingosine kinase transform sphingosine to sphingosine 1-phosphate. You understand? So three inhibitor is the blocking the lipid metabolic pathway from upstream GW to downstream DMS. Okay? So can you explain about this?
Yeah.
Then what is the sphingolipid? What is the difference from the phospholipid? Most popularly known the phospholipid. Can you explain what is the difference between the sphingolipid and the phospholipid? Tamana, can you explain about this? Sorry?
But maybe backbones, there is some difference.
Right.
Backbones.
Backbone is different, totally different. Some difference, not some, totally different. So what is the backbone of a phospholipid?
Glycerol backbone.
Right, glycerol. What about the sphingolipid?
Sphingosine backbone.
No, serine. Amino acid serine backbone. You understand? Okay? Okay. Go ahead. 죄송합니다. 제가 너무 딜레이 해가지고. 너무 설명이 부족한 것 같아가지고. Okay, go ahead. 임난.
The sphingosine kinase inhibitor majorly reduce the release of the exosomes, and it also reduce the interleukin-1 beta activation in the macrophages.
When treated to macrophage, right?
Yes. And there are two kinds of sphingosine kinase 1 and 2. And it was found, and when the cells knocked down with sphingosine kinase 1 and 2, it was found that the knockdown of the sphingosine kinase 2 significantly impact the release of the exosomes from from the cell, and same when these both were overexpressed in the cells, the sphingosine kinase 2 had major impact on the release of exosomes when compared to the sphingosine kinase 1, and it concludes that the sphingosine kinase 2 is involved in the S1P mediated release of exosomes from dying cells. And when the cells were treated with staurosporin and uh, 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 and pre-incubated with S1P, it was it 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 increased the amount of exosomes released when compared to just staurosporin treatment to the cells. And then it was and then confocal in confocal microscopy, as sphingosine kinase 2 remained close to the plasma membrane in earlier apoptotic phase, but not sphingosine kinase 1. Presence of sphingosine kinase 2 near plasma membrane in early apoptotic phase contributes to the increased regional sphingosine 1 phosphate, which causes formation of complex fine spindle-like structures at plasma membrane, and then subsequent maturation of the CD63 plus vesicles. And then S1PR1, there are 4 kinds of sphingosine 1 phosphate receptors, namely from sphingosine phosphate receptor 1 to sphingosine phosphate receptor 4. So the inhibitors for the sphingosine 1 phosphate 1 to 4, the antagonist of these receptors reduce the release of exosomes. Only the S1PR3 and 2 antagonists led to reduce the AV exosome secretion, but the others did not. And then the role of S1PR1 and S1PR3 was found that in the confocal microscopy, the S1PR1 and S1PR3 were co-localized with the EGFP CD63 particles when shown in the when after the treatment of the staurosporin, and it was also confirmed by the Western blot that the expression of S1PR3 was in 3 and 1 was increased in the exosomes, while the S1PR2 and S1PR4 was not significantly expressed on the exosomes. And same was confirmed by the flow cytometry. And in this experiment, it was tried to find that what component of the G protein coupled receptor is involved in the formation of AEVs.
None.
Yes.
You need to explain some information, pre-information. Without information, you abruptly use if you discuss about the G protein, no one understand about this. How can you other people understand about G protein? Why next experiment we searched about G protein, role of G protein in exosome biogenesis? Why?
The sphingosine 1 phosphate receptors are G protein coupled receptors.
You need to explain about that. Without any explanation, no one understand about this. Okay? Okay.
Hence, the sphingosine 1 phosphate receptors are the G protein coupled receptors, multiple G protein coupled receptor inhibitors were tried to find out that which subunit of the G protein is involved in the release of AEVs, and for that, the one inhibitor was pertussis toxin, which is for GI alpha, and
None. Without any information about G-coupled receptor, how can other people understand about this? Impossible. Can you explain about how the G protein coupled receptor activation transduce downstream? Can you? No? No? You cannot? Okay.
Imran, can I ask you one more question?
yes,
Do you know how the other protein move into the actin cytoskeleton? Do you know how to do it?
other proteins.
Yes, you know how the actin cytoskeleton looks like in the cell. Do you know?
I'm sorry, I don't understand your question.
So, do you know how cytoskeleton looks like in the cell?
내일도 도와줄 수 있긴 해. 아마 12시간부터 2시간
Some, it's like expanded fibers.
Yes, cytoskeleton.
Filamentous
How the
structure.
cytoskeleton looks like in the cell?
I'm afraid I don't.
12시간?
The reason I'm asking this question, the most of the transporter or protein signaling relate to the cytoskeleton signaling with they move they're more like a spider web inside the cell. So from that cytoskeleton they move to the surface or outside, or they create the signaling and everything. And that's why we check the beta actin, because it's the structure the component of the whole structure in the cell. So let's say it's like modulation of actin skeleton. It means not just like you're triggering signal and then they floating around the cell. They're just using the network like the spiderweb network, and they try to up and down and create the signal. So that's why I'm asking. That's why the Professor Che asked for the couple the G protein and how is the subunit plays a key role in the formation of AEB. Do you understand? So you have to look for the why this signal it's not just every signal is just not floating around. Okay, they have to through with the couple with the cytoskeleton structure itself. Okay, cool.
Multiple G protein subunit inhibitors were used, and the pertussis toxin is the inhibitor of GIA, and YM25 is the inhibitor of the GQA, and Y27 is the inhibitor of G1213A, and gallin is the inhibitor of G beta gamma subunit. And it was found that the inhibition of the G beta gamma subunit reduced the release of exosomes, while others did not have the effect.
Okay, I need to explain briefly about the G protein coupled receptor. Okay, actually, G protein coupled receptor is very, very important plasma membrane receptor. Actually, I remember the more than 1,000 GPCR exist at the plasma membrane. Very, very important receptor. And then firstly found signal transduction is from G protein coupled receptor. The founder of GPCR getting Nobel Prize. Very, very important. Okay. G protein coupled receptor usually does not have any signaling molecule at the plasma downstream at the cytosolic phase of plasma membrane. You understand, Sheba? Right? So when the ligand binding GPCR gather together, right? So make aggregate plasma membrane aggregate. Then at the cytoplasmic part of the GPCR, recruiting G protein, G protein, G protein. So three kinds of different G protein exist: alpha, beta, gamma. Usually, alpha and beta gamma acting the individually, independently acting. But anyway, activate GPCR, recruit G alpha and G beta gamma. Okay, okay. Then you mostly known the popular signaling pathway caused by G alpha subunit. Actually, depending on GPCR, the specific G alpha subunit recruited, because G alpha subunit have some various kind of different protein. For example, GI, GI and G1213, and then GQ, right? You understand? So the pertussis toxin is an inhibitor of GI. And then YM blah, 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 I remember the inhibitor of G1213? GQ? Okay. And Y blah, blah, blah is the inhibitor of the ROC. ROC is a downstream the pathway signal from the G1213. So when the G alpha signal, all kinds of G alpha signal, which suggested the transduce signal from G S1PR1 or 3, when all the inhibitor shows no effect in the exosome release. That's why I'm thinking about beta gamma. Around 2028, when I'm looking for the paper, some small JBC paper suggests G beta gamma also can transduce signal. Okay, and they recommend suggest G beta gamma mobilize actin cytoskeleton, cellular actin cytoskeleton. Okay, so the very, very interestingly, the inhibition of G alpha subunit shows no effect. But when the gallane is the inhibitor of G beta gamma, gallane almost all block the exosome biogenesis. That's why and then next, Imran. Oh, Imran, you did not show the confocal images. Okay. So,
And then
so
I'm
we
sorry.
already know the CD63 the mobilization in during the cell death, right? So the actin cytoskeleton is also co-stained by the phalloidin. Phalloidin is the phalloidin is specific the binding partner of effectin, filamentous actin. Okay, so the red fluorescence conjugate phalloidine exactly bind to the F-actin, cellular F-actin. And then what is the result of the F-actin?
It shows that the CD63 positive particles are co-localized with the filamentous actin, and after 4 hours they are disconnected as they move to the perinuclear region where they are surrounded by the vacuoles and then ultimately released by
Slowly.
the cell.
Mhm.
And when F-actin inhibitor cytokeratin D was used, and it was shown that the inhibition of the cytokeratin D inhibition of the filamentous actin also resulted in the reduced release of exosomes when subjected to the apoptotic cell death. And then to find out that what part of the exosome activates the macrophages, the exosomes were incubated with protein kinase, RNase, genase, and Triton X100, and it was found that the co-treatment with Triton X100 reduced the activation of the interleukin-1 beta, while others did not. It shows that the some part of the membrane, which can be either a lipid or some other part, has a role in the activation of the macrophages, and just the whole exosomes and also the exosome membrane was checked for the acquisition of exosomal markers, and it was found that they were highly expressed in the membranous part. Also, the membranous part has greater tendency to uh, uh, induce the inflammation in the uh, uh, macrophages. And when the exosomes pre-incubated with the sphingosine 1 phosphate showed greater induction of the inflammation when compared to the just exosome or just sphingosine 1 phosphate.
So what does it mean?
It means that the sphingosine 1 phosphate is responsible for the activation of inflammation in macrophages. And then the G protein subunit inhibitor.
SMPR1, SMPR inhibitors.
Sorry, the S1PR inhibitors were used, and it was found that the S1PR3 inhibition reduced the interleukin-1 beta activation, while others did not. And
Overexpression of SMPR1 or 3.
yes, overexpression of SNPR1 and 3 greatly induced the induction of inflammation in the macrophages. And then the G protein subunit inhibitor like CCG and Rho-A inhibitors were used along with the G protein coupled subunit inhibitors, and it was found that the
Actually, the in this experiment actually incomplete, but data showing the actually sphingosine 1 phosphate receptor SMPR1 and especially 3 highly expressed at the exosome. Everybody knows in these days we are using the sphingosine 1-phosphate receptor 3 as a cell death associated exosome EV, right? So these results showing that the sphingosine 1-phosphate receptor with their ligand sphingosine 1-phosphate collectively activate macrophages and induce inflammatory cytokine, for example, IL-1 beta. And this signal can possibly caused by the node 1-2 signal, one of a pattern recognition, intracellular pattern recognition receptor. Okay, next slide.
And here, it is confirmed that the apoptotic exosome vesicles have DAMPs, as the apoptotic exosome activated macrophages. The RNA was collected from them, and it was sent for the RNA sequencing, and it was found that the most of the upregulated genes are involved in the cellular inflammation, like interleukin-1 beta or NOS2, COX-2, and others. And here it is shown that how many genes are involved with the NF-kappa B or the MAP kinase pathway, and the maximum amount of the upregulated genes are involved with the NF-kappa B. And as it is previously confirmed that the S1P works by activating the NF-kappa B for the activation of inflammation in the macrophages. And when it was checked for the gene ontology and the biological process, the most of the genes were involved with the immune system or innate immune response, and most of the cellular component was either the membrane or the endoplasmic reticulum or the cytoplasmic vesicle parts. And most of the molecular functions were either protein binding or chemokine receptor binding. And in discussion, it is schematically shown how the S1P is involved in the release of exosomes. In the early phases, sphingosine kinase 2, which catalyzes the sphingosine 1-phosphate. Sphingosine 1-phosphate binds with the S1PR1. S1PR1 translocalized on the membrane, which when the S1PR1 is activated, the activated receptor transduces a signal for the provocation of the actin polymerization. The actin cytoskeleton then enhances the formation of endosomes, which then mature into the multivesicular endosomes, and the multivesicular endosomes then move to the cell membrane, and from there the exosomes are released. Yes.
I made another schematic diagram.
Yes.
Okay.
Imran, can I give you some comment before I ask a question?
Yes, please.
Okay. For my understanding, I have some knowledge on the immunology because I already learned some of the immunology part, and then I understand what you're trying to say. But thing is, when you show the result, I think you have to put some legend. Why you have to show like there's the some title, but you have to like add some legend why you're doing this experiment, and then you have some inhibitor and everything. You have to show that what this kind of inhibitor have different function, and because of this, I don't really know what you're trying to say. Okay. And another thing, there's no as the Professor Che showed, there's no schematic. You have to before you showing the whole result, you have to add a whole bunch of background first from the introduction. And for if you have a more time, when you show the result, according to the result, you have to draw some signaling picture for drawing. Okay. For this one, I even you when you even you're trying to explain the result, I have to look for the whole thing at the time. What you're trying to say? You're just showing the whole result, and that's it. Because that means the most of people who doing this journal study don't some of them have the knowledge, some of them don't have knowledge, some of them even don't know what's the exosome or endosome or anything. They don't even know that what the dumps means. Okay, you have to explain like key keyword, like key abbreviation, which is this word is this word is this. Okay, and I want to ask a question. For this journal paper, it's done only by macrophage for relating to information. What about other AP cell like monocyte or dendritic cell or eosinophil? Do you think have the same result when you do the experiment with these immune cell?
I hope there will be similar results when tried in other monocytes.
So as you know that the cell I mentioned about, they have a CD63 and that they're related to interleukin 1B. And do you think it's the same result or different result?
I
If
think
you
same
like
results.
the using AAV to targeting for endosome, do you think it's the same result?
Yeah.
Maybe not, maybe. Actually, in this paper, the uh, contained also in vivo data, right? In vivo study, in vivo. Did you read that?
Mhm.
In vivo.
Yeah, there's NOD mouse that you
Uh,
trying to the top pages there you're showing the result, right?
Actually,
Yeah, I saw the data is done with NOD mice.
난 actually 제가
저기 아까 NO 마우스, NOD 마우스 있으면 NO 마우스인가요? NOD 마우스인가요? 저
NOD
위쪽에
아니고요. 그
아닌가요?
노드 시그널이라고
음.
패턴 레코그니션 인플라마토리 시그널 정원하는 인트라셀룰라 리셉 패턴 레코그니션 리셉터예요. 노드
아, NOD라 그래서 저기
그
너카웃 마이스를 쓴 줄 알았더니
아니
그게 아닌가.
마우스 아니고
응.
이거 인비보 실험은 마우스에다가 아마 엑소좀을 준 다음에 인플라메이션이 어떻게 간이나 뭐 혈액 내에서 뭐가 보이고 뭐 그런 거 했을 거야 아마. 그다음에 뭐 저기 스타우로스포린을 때렸나 뭘 LPS 줬구나. LPS 같은 거 주고 셀데스 유도한 다음에 엑소돔 이런 타입의 에이포프토틱 엑소돔 분리하는 거 보고 그랬을 거예요 아마. 그다음에 인비보 마우스 엑소돔 주고선 그 인플라마토리 사이토카인 익스프레션 내는 거 봤을 거예요 아마. 안 보려고 그랬는데 그 저기 리뷰가 보라 그래가지고 아마 했을 거예요 아마. 불이 나게 했던 것 같고. 그 사실 이 뒷부분은 제가 아주 지금도 잘 모르겠고 이거를 해결하려고 제가 지금도 많이 노력하고 있는데, 그래가지고 최근에 우리 송다정 학생이 요거를 반복을 해서 하는 거죠. 왜냐하면 첫째, 이때 뽑은 엑소점이 퓨리티로 따지면은 그렇게 좋았다고 저는 생각하지 않아요. 이때는
네,
처음 하던 시절이래 가지고
네.
그 퓨리티가 별로 안 좋았다고 생각해요.
그러면 여기서 진 데미지도 같이 일어난다는 가정하에서 하는 건가요? 지금 어떻게 된 거예요? 보니까 저기 노말하고 진 데미지 일어난 거가 그렇게 있는 것 같지가 않은데 이게 진 모디피케이션 자체가 저 디스트럭션에 관련된 모디피케이션이지 아니면 그 자체에서 저 핵에서 이게 그 인듀싱이 되는 건지 굉장히 지금 잘
아,
모르겠고 그게
그
잘
엑소점 바이오제네시스
모르겠어 지금 돼
이게
있어서 이게 그 모디피케이션의 단계가 이게 그 에이팝토틱 하면서 이제 하는 건지 아니면은 아예 그 크로모존 모디피케이션을 일으키는 상태에서 하는 건지 저도 확실하지가 않거든요. 이렇게 봐서는.
그 크로모솜하고는 그렇게 관련이 별로 없고요.
네,
그 세포 사멸이 일어나는 과정 중에
네.
그 EB가 합성되는 과정이 있는 것 같아요.
음,
핵하고는 따로 노는 거고요. 크롬
그러니까
모디피케이션하고는 별 관계가 없고,
그럼 어디에 들어가는
아,
거죠?
예?
플라스미드 DNA인가요? 아니면 저기 그쪽에 들어가는 건가요? 아니면 마이토콘드리얼 DNA가 들어간 건가요?
저이 덴프로스 역할이요.
네,
그거를 잘 모르는데,
네,
그 DNA나 RNA는 아니고요.
네,
그 스핑고신 1포스페이트하고 그다음에 스핑고신 1포스페이트 리셉터가 그 엑소존 멤브레인에 많이 자리하고 있거든요.
네, 네, 네,
이 리셉터가 자체가 그, 그 매크로파지에 가가지고
네.
얘가 신호를 전달하는 것 같아요. 따로.
음,
그래서 NF-kappa비를 활성화시키고 그다음에 예. 근데 정확한 기전은 모르겠어요. 그러니까 DNA나 RNA에 의한 덴프로스 그런 건 아니고 이 멤브레인에 있는 리셉터하고 스핑고신 1포스페이트가 자극하는 거는 맞는 것 같아요.
이대로 하면은 그 오토이뮤 디지즈 쪽도 관여가 있을 수도 있겠네요.
어, 당연히
그럴까요?
관여할 수도 있을 거라고 생각을 합니다. 아,
지금 기전이 좀 비슷한 것 같아서
기존요?
네,
오토이문
오토이뮤 일어나는 인플레이메이션에서 공격하는 패턴이 비슷한 거.
그렇죠,
예,
그렇죠.
예. 그래서 비슷한 것 같아가지고 지금 그런
그렇죠.
거였는데, 음,
비슷할 수도 있죠. 예.
음.
제가 또 요즘에 관심 많아가지고 공부 많이 하고 있는데요.
네네네.
아직은 잘 모르겠습니다.
아니,
어,
그래서 저도 그 기전이 좀 비슷한, 그, 나중에 지금 공격해서 인포메이션 일으킨 패턴이 이거랑 비슷한 것 같아가지고 그런 것 같은데,
예. 그, 그래서 사실은, 어, 여기 보면은 스핑고신 1포스페이트 리셉터가 되게 중요하거든요.
음,
그게 좀 노블한 건데, 그래서 최근에 그 엑소존 뽑는 랩들 있잖아요.
네네네.
랩들에서 쓴 리뷰 페이퍼를 보면
네,
이 사람들이 본 이유를 알고 쓰는지 모르겠는데 스핑고신 1포스페이트 리셉터를 갖다가 스핑고신 1 그 포스페이트를 셀
네.
컬처하면서 주면서 컬처를 하더라고요.
음,
그거 갖다가 프라이밍이라고 얘기를 하대. 근데 상당히 일리가 있는 얘기라고 생각해요.
여기,
이거만
네.
보면은 스핑고신
그,
1
여기 보면은 G 프로테인, 베타 감마 같은 경우 이게 어비니티 테스트는 안 한 것 같네요. 얼마나 있는지, 그거 가지고는. 그냥 그, 이게,
G 프로테인 시그널이요?
네, 시그널 어피니티
그,
테스트는 안 한 것 같은데 그 에세이는 없는 것 같던데요, 보니까 있었나요?
예, 여기, 이게 막판이라 가지고 그것까지는 안 해봤고요.
네,
예, 예.
음,
그런데 요즘에 제 생각은
네,
여기서는 억지로 액틴 패스웨이가 관여할 거라고 그냥 그 결론을 몰고 갔는데 요즘에 나오는 페이퍼를 쭉 읽어보면 그 그 베타 감마에 의한 액틴 폴리머라이제이션, 액틴 사이토스켈레톤이 생기는 것보다는 그 이 스핑고신 1포스페이트가 리셉터가 엔도사이토시스 되는 게 중요한 것 같아요. 그쪽으로 생각을 사실은 임란이 열심히 공부해가지고 그걸 좀 알아가지고 여러 가지로 실험을 많이 할 줄 알았더니 이 3년, 2년이 되도록 이 페이퍼도 이해를 못하고 우리 있어가지고 실망해가지고 읽으라고 그랬는데 예, 지금 제 예상으로는 그 스핑고신 1포스페이트 리셉터가 활성화되면 그 엔도사이토시스 안으로 들어오는데 그게 엑소전 바이오제네시스하고 관련이 있는 것 같아요.
지금 보면은 제가 아까 보면서 사이트 스캔 거에서 거기다가 사실은 저 플로렌스 태그 붙여가지고 하면 이게 사이토 스켈레톤 저기 네트워크를 돌아다니니까 실시간으로도 그 시간별로 측정이 가능할 것 같은데.
있는 Do you remember the live actin monitoring probe? What we call that? I gave you the plasmid,
Yes.
the staining F-actin, the protein with a high affinity to F-actin. You know, we call that what? 그래가지고 저도 F-actin,
Life Act.
앞소리?
Life Act.
라이프 액트라고 있어요.
아,
여기서는 팔로이딘을 써가지고 F-actin을 스테이닝 했는데요.
네,
이거는 죽은 세포 팔로이딘 자체가 독소래 가지고
네,
죽어 있는 세포밖에 못 하기 때문에 이거 라이브 이미징 하려고
네.
아이팩트를 그걸 해가지고 해보려고 그랬는데 아직까지 임란이 시작도 못 하고 있어요.
보면 제일 그렇게 하면 그 왔다 갔다 네트워킹 형식으로 퍼렇게 빨갛게 위에서 저기 붙은 다음에 곧바로
그런데
네트워크로
그런
볼 수
리피드
있을 것 같아요.
프로브라고
응,
하는데요. 리피드
음,
바이오센서
음.
프로브라고 하는데 어, 그렇게 아직까지 기술상으로 굉장히 완벽한 프로브가 없어요.
그러니까 제가 했던 것 중에 하나가 저기 박사 때 했던 것 중에 하나가 글루코스 트랜스포터에서 이제 믹 에피톱을 붙여가지고 형광물질 붙여서 이제 실시간으로 했던 그게 있었어요. 저희 쪽 랩에서. 그래서
예,
예.
네, 저희도 지금 하고 있어요.
음,
다음 페이퍼는 이 엔도사이토시스 된 다음에 멀티베스큘러 엔도솜을 만드는 과정에 대한 연구인데요.
네네네.
그거 열심히 해가지고 대박으로 실패했어요. 있는 돈 없는 돈 다 때려박아가지고
그거
한
만만치 않죠.
엄청 많이 해가지고
음.
그 데이터가 막 완전히 인간이 실망스러운 건 인난 in this paper lot of more supplementary the data but
Yes.
you did not explain about this. So I'm very, very disappointed. 2 years, 2 years you stayed in our lab in 2 years. You still do not understand. Then I completed and also did not completely read the paper. It's very, very basic paper for your further study, but you did not. Something wrong. Imran, not good. Okay? Huh? So in next class, at our present time, please the present the uh, the following paper about gestamine E. Okay, okay, okay. 음. 근데 뭘 얘기하다가 갑자기 화가 나가지고.
저기 네트워킹 해가 저기 뭐지 그 사이트 스킬 네트워킹에서 이렇게 왔다 갔다
그래가지고
응.
저는 사실 여기서 더 궁금한 게 그 에펙틴도 중요하지만 그 리피드가 되게 중요하다고 생각을 해가지고요.
네네네.
리피드 센서 같은 거 해가지고 실험하려고 계획은 짜는데 아직 시작도 안 했어요. 음. 임난, do you remember we bought several lipid sensor for we can use at the live imaging. You remember?
Yes.
네. 그래가지고 그거 하고 있어요. 그래서 이게 여기서 오늘 사실은 제가 재밌는 페이퍼를 읽었어요. 그 뭐냐면 actually everybody knows the tetraspanin such as CD63, CD81, CD9 is a very very famous exosomal marker. Sheba, Tamana, where is Tamana? Sheba, you know, Imnan, you know, exosomal marker, tetraspanin. But actually, in this type of cell death associate EV, also the CD63 is a very, very highly expressing marker, right? You showed us about that, right? So my question is that why CD63 or the CD9 molecules are highly enriched in the exosome? Why? Why? Because I read today I read a very very interesting two paper from the Stephen Gould, Stephen Gould of in the Johns Hopkins University. Actually, his research exactly same with me, right? Sheba, did you read? But actually, his the mainstream of research was that the CD63 plasma membrane localization is very important for the exosome-sized vesicle biogenesis. That means when the CD63 the highly enriched in the plasma membrane, then the CD63 expressing the EVs is very portion of the CD63 EV is very very highly increased. So what do you think about that?
Like why CD63 is present on
Why the plasma CD63 expression in plasma membrane is important for the EV exosome-sized exosome-sized vesicle biogenesis?
because CD63 is a tetraspanin protein and it is involved in the trafficking and cargo.
I can understand.
Because CD63 is a tetraspanin protein, is a transmembrane protein, and it is highly enriched in the late endosomes and the multivesicular bodies.
So,
and the exosomes arise from the multivesicular bodies or the late endosomes. That's why CD63 is prominent in exosomes.
The existence of CD63 in the multivesicular endosome does not mean the CD63 is important for the exosome biogenesis. Okay, the Dr. Stephen Gould's research is mainly focused on when she modified cells, the CD63, endosomal CD63, when he made the endosomal CD63 to express at the plasma membrane, then exosome-sized vesicle contained a lot of CD63. Always same. CD9, when the cell modified CD9 or CD81, expressed highly at the plasma membrane. Then the exosome-sized vesicle enriched in CD9 or CD81. That is exactly with my data. Do you remember? Before the starting of the cell death associated EV, CD63 and SMPR1 previously or formerly exist as a cytosolic vesicle, they abruptly move to the plasma membrane. Do you remember? Hmm? So this kind of cell death associate the exosome biogenesis. All the SMPR1 movement to the plasma membrane is first step. Okay? We reconfirmed in the JI paper in 2023. Okay? And then at the plasma membrane, the SMPR signal-based endocytosis is a first step for biogenesis of exosome. You remember? Okay. So I guess CD63 and then CD9 or 81 is just bystander. Do you mean what I mean? Bystander effect. CD63 just near SMPR103. So during the endocytosis of SMPR, they together they mobilize to the exosome and endosome. So the during the maturation of MBE, SMPR1 containing cell death associated exosome contain SMPR1 the CD63. Can you understand? Bystander effect. Bystander. You understand? Really? So so based on the bystander effect of something, when we change the plasma membrane of the distribution of a sphingosine 1-phosphate receptor, then exosome cargo loading, including SMPR1 or 3, will be totally changed. You understand? Then this hypothesis corrected with what? What? So I believe the drug, the inhibiting the PI45P2 or cholesterol, making the change in plasma membrane distribution of a sphingosine 1-phosphate receptor. You understand? This is my hypothesis. Okay, hypothesis. For the, sharing this hypothesis with you in 2 years, every time when I reading new paper, I send you the paper, but you never understand. You understand? Eh? That's why I'm very, very angry at you. You understand? Eh? 죄송합니다.
괜찮습니다.
네.
오늘 하고 you did great job, Imran. Okay. 그 다음 주에 저희 쪽 김예원 학생이 지금 저희 하는 프로젝트 관련해가지고 아마 발표 진행을 할 거고요. 아마
네.
아직 페이퍼로 나온 상태는 아니어서 잠깐 저희가 좀 숨기고 있는 상태라서 그 내용에 대해서 아마 발표를 할 겁니다. 예은 학생 다음 주 화요일 날 준비하는 걸로 하고 오늘 마무리하고 그리고 Imran, would you please share your journal paper also? I'll I there's my lab email address I can send to Professor Che and you can send the journal paper to me and I can spread out to everyone. Okay? 오케이. 수고하셨고요. 다음 주 화요일 날 동일 시간에 다시 하겠습니다. 진행하겠습니다. 그리고 이거는 계속 유튜브에 업로드 하니까 보시면서 다시 리마인드 학습하시면 좋겠습니다. 그럼 수고하셨습니다.
감사합니다.
수고하셨습니다.